A very warm good evening, guys. Good evening to all. Today we are having yet another lecture from one short series from your favorite and my favorite BioPoint Stream the World of Bio. So now we are going to start with Cell, the Unit of Life. We haven't done the last part of this chapter, and today we are going to complete the entire chapter in one shot. Whether the one shot will be part two, whether the one shot will be having part two or not, we will complete it surely, because this presentation contains almost 74, yeah, exactly 74 slides, and we should complete each and everything in one session. Okay, if we can't do that, surely we will be doing it as a part two. So okay guys, just move forward in this session on cell, the unit of life. So once again, welcoming you all to the one-shot session on the chapter Cell, the Unit of Life. And for more updates, join our Telegram group t.me/bypointnew. Now we have started the yet another series of one shot for the NEET 2021 and 2022 batch. Along with the one shot series, we will be continuing the old sessions, regular sessions with that too. In order to have a constant touch with the portions, we are doing one shots too. After that, we will be back with a question discussion, aim at 100K now, which we are doing it in the form of polls in our Telegram group, World of Bio and 2021 and 2022 group. So, guys, hope you all are ready for yet another session from BioPoint, Stream the World of Bio. So, let's move forward. Once again, remembering you, do not forget to hit the like button, do share it with your friends, do subscribe our YouTube channel, BioPoint, Stream the World of Bio, and do not forget to remark us by leaving a comment in the comment section. So, hope you all are perfectly ready to start this session. If you are ready, give me a thumbs up on the screen. And now we are going to start the yet another session on cell, the unit of life. Cell, the unit of life. So the topics that we are going to cover in this one short session are what is a cell and the cell theory, an overview of the cell, then about prokaryotic cell, that the cell envelope, its modifications and ribosomes and inclusion bodies. And if it is in case of eukaryotic cells, we are going to describe about cell membrane, cell wall, endomembrane system, mitochondria, plastid, ribosome, cytoskeleton, cilia and flagella, centrosome and centrioles, nucleus, and microbodies. Hope you all are thrilled for this session. So let's move forward. Have the Josh in the class. And now we're going to start this session. Introduction. So what is a cell? As you all know, a cell is the structural and functional unit, fundamental structural and functional unit of all living organisms. And a cell was discovered. A cell is discovered by a scientist known as Robert Hooke. Robert Hooke discovered cell. And a scientist known as Anton van Leeuwenhoek first observed the living cell. He was the first scientist to observe a living cell. Clear with that? So a scientist who discovered cell is this Robert Hooke, and the one who observed the living cell, it is Anton van Leeuwenhoek. So moving forward in this session, cell theory. So the very base of the cell theory, it is proposed by two scientists, that is a botanist and a zoologist, that is Schleiden and Schwann. In 1838, Matthias Jacob Schleiden proposed the cell theory, and in 1839, a zoologist named Theodor Schwann added his point to the cell theory. So Schleiden added the points based on the plants, and Schwann added the points based on animals. So these two scientists proposed the cell theory consisting of a single point. All living organisms, whether it is plants or animals, are composed of cells, and all activities in an organism are due to the sum or product of the cell. These are the 2 points that Schleiden and Schwann added. And the last point, all cells arise from pre-existing cells, is added by a scientist known as Rudolf Virchow later. Okay, Rudolf Virchow later added his points in 1855. He added his points by using a term known as omnicellula e-cellula, and thus is how he added his postulate to the cell theory and how he modified the cell theory. And the cell theory composed of all these 3 points is known as the modified or the modern cell theory. Clear with that? The next topic is an overview of cell. An overview of cell. So the size of a cell, if we say about that, it varies from organism to organism, and the shape too, it varies from different cells to cells. So cells contain different organelles present in the cytoplasm of the cell, and it has a nucleus at the center. So that is how outline, if we say about the structure of a cell in an outline, we have a cell contains cytoplasm and ribosomes in case of prokaryotes, cytoplasm, well-defined nucleus, and other organelles, presence of other organelles in eukaryotic cells. Clear with that? Next, what we say about is the size of the cell. So hope it's clear till now. We have discussed about the very base of the cell, the very basic definition of a cell, the cell theory formulated by Schleiden, Schwann, and Virchow. That's a modern form of the cell theory, and we have discussed the very what an overview of a cell. These are the 3 topics that we have covered till now. Okay, I don't know whether I'm moving a bit fast or not. Anyway, hope it's clear till now. The smallest cells are mycoplasma. The smallest cell ever found is the mycoplasma. That is 0.3 micrometer. Size of the mycoplasma is 0.3 micrometer. Okay. Largest isolated single cell is egg of ostrich. Longest cells, nerve cells. Size of bacteria 3 to 5 micrometer. Human RBC 7.0 micrometer in diameter. So the smallest living cell in human body, it is human RBCs. And the longest cell in human body is the nerve cell or the neuron. Clear with that? Based on the shape of the cell, based on this function, shape of cells may be disc-like, polygonal, columnar, cuboid, thread-like, or irregular. So these are the different shape of the cells based on the different what types of cells or different kinds of cells based on the function and shape of the cell. Clear with that? Just leave all those things. That's all are very basic and not even needed for your examination. This is from where we have to focus all our attention. That is the type of the cells into prokaryotic and eukaryotic cells. So cell can be classified into two as prokaryotic and eukaryotic cell. So what do we say about the prokaryotic cell? Prokaryotic cell refers to what? This prokaryotic cell means it has a primitive nucleus, right? Primitive plus nucleus, that is what karyon refers to nucleus and pro refers to primitive. So primitive nucleus is what we say about prokaryotic cell, and eukaryotic cell, if we say about that, it has a true nucleus. That's a true nucleus. So guys, if you all are here, give me thumbs up on the screen. I will just wait for that for the next 3 seconds. Come on, be fast. Okay, then let's move forward with the different type of cells, prokaryotic and eukaryotic cells. So the main point is that well-defined nucleus and organelles are found in eukaryotic cell, whereas in prokaryotic cell, non-membranous organelles are present. Only non-membranous cell organelles are present in case of prokaryotes. Clear with that? So now we are gonna move with start the session on prokaryotic cell. So if we say about prokaryotic cell, no membrane bound nucleus and organelles are found in prokaryotic cells. No membrane bound nucleus and organelles. If the nucleus, it has no membrane and the organelles. So membrane bound organelles, all organelles are membrane bound except what? The ribosome. So the cell organelle present in prokaryotic cell, it is the ribosomes and it is of the 70s type. 70s ribosomes are present in case of prokaryotic cell. So then what is this S refers to? S refers to the sedimentation coefficient. S refers to the sedimentation coefficient. That all we will learn in the upcoming slides. So now just focus. So if we say about the examples of prokaryotic cells, we have bacteria, blue-green algae, mycoplasma, and PPLO. We refer mycoplasma is also known as PPLO, right? Pleuropneumonia-like organism. That is what if we expand PPLO. So generally smaller and multiply more rapidly than eukaryotic cell, vary in shape and size. This is what if we say about prokaryotic cell. Clear with that? Moving on to the detailed part. So prokaryotic cells, now we are going to say about the different shapes of bacteria on the topic prokaryotic cell. So guys, just wait for 2 minutes and revise all the portions that we have covered till now. So is it clear till now? Hope it's fine. Okay, then now we are moving on to the topic. So bacteria, on the basis of their shapes, we can classify it into four: Bacillus, Coccus, Vibrio, and Spirillum. Okay, so rod like Bacillus, spherical Coccus, Vibrio comma shaped, Spirillum spiral shaped. Okay, so these are the different shapes of bacteria which we are gonna learn now. Okay, so just remember rod like Bacillus, spherical Coccus, Vibrio comma shaped, spiral shaped Spirillum. Okay. Oh my god. Okay, so this is an outline of the prokaryotic cells having a plasma membrane, cell wall, plasmid, pili, ribosomes, flagella, and a cytoplasm. Okay, and we can see no, we can see a primitive nucleus or a nucleoid that is just DNA just wrapped around just what DNA just found in circular as threads without any specific outer covering. So now we're going to start with the cell organelles of prokaryotic cell. Cell envelope, mesosomes and chromatophores, nucleoid, flagella, pili and fimbriae, ribosomes, inclusion and inclusion bodies. These are the topics that we are going to start now on the cell organelles of prokaryotic cell. Cell envelope. Cell envelope is chemically complex protective covering. It is composed of tightly bound of three layers: glycocalyx, cell wall, and plasma membrane. Glycocalyx, cell wall, and plasma membrane. So we have to learn all these layers in detail because it is important in the exam point of view. Okay. So glycocalyx, also known as a cell coat, right? Glycocalyx, also known as a cell coat. Plus, plasma membrane, also known as what the new term which I have always remembered you, biomembrane, or as usual, cell membrane. Clear with that? Glycocalyx. Glycocalyx is the outer layer. One second. Glycocalyx is the outer layer. Its composition and thickness may vary in different bacteria. It may be a slime layer, a loose sheath, or a capsule that is a thick and tough layer. So the layers of glycocalyx is classified into two as slime layer and a thick capsule. So the thin layer is known as the slime layer, and the thick layer is known as the capsule. Guys, just don't leave the session seeing that it is premiering. So just focus in the class. I'm seeing you in the chat box. I'm just constantly having a view on that. So just have a focus and comment all your doubts. Surely it will be cleared. No issues with that. Okay. So and the layers of glycocalyx and the layers of glycocalyx are basically made up of polysaccharides. So the component of the cell wall, component of the glycocalyx layer is polysaccharides. Hope the screen is clear. I don't know. It's not a live, so I cannot have a direct communication with you. So hope it's clear, perfect and fine. So cell wall. Next it is a cell wall. It is a middle layer seen in all prokaryotes, gives shape to the cell, provides a structural support to prevent the bacterium from bursting or collapsing. So it is basically made up of the cell wall of this prokaryotes is basically made up of peptidoglycan. We have already discussed in biological classification. Okay, and the first lectures of cell, the unit of life 2. It is basically made up of peptidoglycan, which is of two components, NAM and NAG. Right, N-acetyl muramic acid and N-acetyl glucosamine. Clear with that? So the cell wall of prokaryotes is made up of peptidoglycan or murin. Clear with that? So let's move forward. Plasma membrane or biomembrane or cell membrane. It is the inner layer. It is semi-permeable in nature and interacts with the outside. This is structurally similar to that of eukaryote. So the universal feature in all organisms, the universal feature, it's an AIMS question. Universal feature in all organisms, it is cell membrane or plasma membrane. All organisms, if it has a cell, there is a cell, it contains plasma membrane. Okay. So next we are moving on to the next part, that is the mesosome. So look, glycocalyx, cell wall, cell membrane, and next we have the mesosome. This is formed by the infolding of plasma membrane that includes vesicles, tubules, and lamellae. So these are certain structures present in the mesosomes, vesicle, tubules, and lamellae. So the mesosomes are formed by the infolding of this plasma membrane. Okay, like this, if this is a cell, due to the infoldings, this mesosomes are formed. Clear with that? Next, a nucleoid. So the prokaryotes basically consist of a nucleoid or an incipient nucleus or a genophore. All these are terms for nucleoid because there is a no well-defined covering around the nucleus. The DNA is scattered there. So formed of non-membranous or naked circular genomic DNA, single chromosome or genetic material and protein. Many bacteria have small circular DNA that is known as plasmid outside the genomic DNA. So outside this DNA, it has a circular DNA. Look here. There are certain scattered circular DNA known as plasmid, and it gives some unique phenotypic characters, that is resistance to certain antibiotics to certain bacteria. Clear with that? Hope it's clear till now. Give me a thumbs up on the screen if it's okay and perfect till now. Come on, guys. So we are moving forward with flagellum. These are thin filamentous extensions from the cell wall of motile bacteria. The number and arrangement are varied in different bacteria. Bacterial flagellum has three parts. So the three main parts of bacterial flagellum: the filament, hook, and basal body. The filament, hook, and basal body are the three main parts of the bacterial flagellum. Okay, so this flagella is made up of a protein known as flagellin, made up of flagellin protein. And what is the purpose of the presence of this flagella? The purpose of this flagella is locomotion. Clear with that? So the filament is the longest portion and extends from the cell surface to the outside. So look here, from the cell surface to the outside. So this long part is known as the flagellum. Sorry, yeah, that is known as the flagellum, and the longest part is known as what? Filament. Pili and fimbriae. These are surface structures that have no role in motility. Pili are elongated tubular structures made up of a special protein known as pilin. Pili made up of a special protein known as pilin. And their function is to help in conjugation by acting as a sexual attractant. By acting as a sex attractant. Clear with that? Fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they help to attach the bacteria to rocks in streams and to the host tissues. Clear with that? It's all about, so to anchorage to the substrate and to attach the bacteria to any outside host surface is known as what? Is host surface is the function of what? Fimbriae. Clear with that? Ribosomes. They are associated with plasma membrane of prokaryotes. They are about 15 nanometer by 20 nanometer in size. Guys, hope it's clear. I'm moving bit fast, but I'm moving with each and every topic, making each and every topic clear. Right? I hope so. Be active in the comment section, guys. Give me thumbs up on the screen if it's clear till now. Any portion, if you have doubt, you may comment it. Surely I will be retaking it live for you. Not as premiering, it as live for you. The part which is not clear for you. So they are made up of two subunits, 50S and 30S. They together form the 70S prokaryotic ribosome. Never add 50S ribosomes, 50S unit and 30S unit to get the 70S. No, never. It's not equal to 70S prokaryotic ribosomes. So just understand this 50S and 30S ribosomes together constitute this 70S ribosome. S refers to sedimentation coefficient, that is a measure of density and size. Okay. What is sedimentation coefficient? It's a measure of its density and size. So what are the functions of ribosomes? They are the site of translation or the protein synthesis. Several ribosomes may attach to a single mRNA to form a chain called polyribosomes. Ribosomes of polysome translate the mRNA into proteins. So they are the site of translation. That is the site of protein synthesis. Several ribosomes may attach to a single mRNA to form a chain of ribosomes known as polysomes or polyribosomes, and ribosomes of a polysome translate the mRNA into protein. Clear with that? Next is about the inclusion bodies. These are non-membranous, stored reserve material seen freely in the cytoplasm of prokaryotic cell. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria, phosphate granules, cyanophysian granules, and glycogen granules and gas vacuoles, etc. These are the components or the structure or the your components, yeah, examples of inclusion bodies. Okay, So inclusion bodies means a structure where the reserved food are stored. Okay. And the gas vacuoles in blue-green algae, purple bacteria, sulfur bacteria, all those help in buoyancy, help in their buoyancy. Clear with that? So that's all for prokaryotic cell. So what we have discussed is about all about this. Look here, we have discussed about the prokaryotic cell. Then we moved on to the classification of different types of bacteria. After that, we moved on to prokaryotic cell, certain a basic overview of its structure. Then we moved on to the cell organelles, that is started with glycocalyx, cell wall, plasma membrane, mesosome, nucleoid, flagellum, pili and fimbriae, ribosome, and its functions, then in inclusion bodies. These all are all about this prokaryotic cells. Clear with that? So guys, just once again have a recap on this prokaryotic cell. We have discussed that there is no membrane-bound nucleus in organelles, bacteria, blue-green algae, mycoplasma or PPLO. These are the certain what examples of prokaryotic cell. Generally smaller and multiply more rapidly than eukaryotic cell, and they vary in their shape and size. Okay. So there may be of four types. Bacteria may be of four types based on basis of their shape: Bacillus, Coccus, Vibrio, and Spirillum. And this is an overview of the structure of a prokaryotic cell. These are the organelles that we have discussed today on prokaryotic cell, about the glycocalyx, about the slime layer and the capsule, about the cell wall and its function that gives shape to the cell, provides structural support to prevent the bacterium from bursting and collapsing. Hope it's fine. Guys, are you able to follow? Okay then. Plasma membrane or biomembrane inner layer, semi-permeable nature, interacts with the outside mesosomes. It is formed of infolding of plasma membrane, includes vesicles, tubules, and lamellae. Nucleoid, an incipient nucleus or a genopore having a non-membranous circular DNA and protein. Many bacteria have circular DNA or plasmid outside. Very sorry, outside the genomic DNA. Flagellum. This is a flagella or flagellum. It is composed of three parts: a filament, hook, and basal body. The protein made is flagellin protein is the protein made composed of what constituting the flagella, and its function is locomotion. Pili and fimbriae. Pili act as a sex attractant, and it has a special protein called as pili and fimbriae to anchorage to the substratum. That's a function of fimbriae to attach to the host tissues. Ribosomes, having 70s type ribosomes of two types, that's 50s and 30s, together forming the 70s. S refers to the sedimentation coefficient. And what is the sedimentation coefficient? It is the measure of density and size. Their functions, that is site of protein synthesis, and we have discussed about polyribosomes and inclusion bodies, gas vacuoles and their function that helps in buoyancy. That all are the things that we have discussed today. Once again, reminding you, do not forget to hit the like button. Do share it with your friends. And if you are a new member, do not forget to subscribe our YouTube channel, BioPoint, Stream the World of Bio, and remark us by giving a comment in the comment section. Thank you all. Thanks a lot. Thank you all. It's me signing out from your favorite and my favorite BioPoint, Stream the World of Bio. Once again, reminding you, just have a focus in the class. Be active in the comment section. Surely we will be there to clear all your doubts. And I'm little bit busy anyway. Abhisar is taking all the portions for you. So if whenever I'm getting time, I'm just rewinding all those portions, and I'm not gonna start with new chapters because or else it may not be getting completed. That's why. So anyway, it's me signing out from your favorite and my favorite Bio Point, Stream the World of Bio. Bye bye.